So our next speaker, Dr. Yiouli Ktena from Johns Hopkins, I'm pleased to introduce her as last year's 2023 CCF Giant Food Next Gen awardee. So congratulations on that. And this is a really important award because back in 1995, wow, way back in 1995, before we called these Next Gen Awards, I was fortunate to receive support from CCF, which helped transition from my fellowship into a faculty level, and that's what these positions are supposed to be. So we're really excited that Giant supports these, and congratulations on getting that. And as part of your potential transition into a faculty level position, you're going to tell us all about donor T-cell DNA methylation in graft-versus-host disease and the graft-versus-tumor effect after allogeneic hematopoietic stem cell transplantation in one breath.
Nice. Strong work. Hi, everyone. Thank you very much for having me here today, and a great big thanks to the CCF for the Next Gen Award. My name is Yiouli Ktena. I'm an assistant professor at Johns Hopkins, and that transition to assistant professor was in part supported by the Next Gen Award, so a great big thanks. I have no disclosures for you today. And we're going to go over three concepts today. We're going to talk about bone marrow transplant and what it is. We're going to talk about graft-versus-host disease and graft-versus-tumor activity. And then we're going to talk about, a little bit height-challenged, I know. And we're going to talk about DNA methylation and how it controls graft-versus-host disease and graft-versus-tumor activity. So when we talk about bone marrow transplant, what we're really referring to is hematopoietic stem cell transplant. So the stem cells, not those that make a whole human, but the stem cells that will make your blood and immune cells. And you can find these stem cells in your bone marrow cavity, that's in your long bones mainly, in your pelvic bone. And if you think of that cavity as the factory, the stem cells are the factory workers that will go on to make your red cells that give you carry around oxygen and give us energy, our white cells that provide immunity, and the platelets that prevent us from bleeding. And so how does a bone marrow transplant work? It sounds like a surgery, but it's in fact not a surgery. We first identify a donor and we collect the stem cells either from the bone marrow cavity directly or from the peripheral veins from their blood, or sometimes when a baby is born from the cord blood. And then the patient will undergo what we call the conditioning regimen. So that's a round of chemotherapy or radiation. And the purpose of that is twofold. It will eradicate any residual disease that the patient may have, but it will also make space for the new immune system, because otherwise the patient's own immune system would just reject the new stem cells. And after that conditioning, the stem cells are infused in the vein, just like a blood transfusion. So it's a little bit anticlimactic, but the magical thing happens where the stem cells know to find their way into the bone marrow and sit there and grow and provide the patient with a whole new immune system that will also help eradicate their cancer. So when we talk about hematopoietic stem cell transplant, it comes in two flavors. So autologous is where we take the patient's own stem cells, collect, save them, and then reinfuse them after chemotherapy. But traditionally, what we're talking about when we talk about stem cell transplant is an allogeneic stem cell transplant, where we get stem cells from a donor, either related or unrelated. And it is still the only curative option for many children with high-risk refractory or relapsed leukemias and lymphomas, as well as for an array of non-malignant conditions. However, graft-versus-host disease remains a major limitation. So what is graft-versus-host disease? It is when the T cells of the donor get infused into the patient and recognize that they are in a foreign body. So they get activated and they start attacking the patient. Nearly every organ can be involved, it can be fatal, and it is always a risk unless the donor and the recipient happen to be identical twins, so have the exact same genes. And a little bit of the pathophysiology of graft-versus-host disease, the conditioning regimen causes diffuse damage and inflammation, because we said it's chemo or radiation, that's what it does. And that releases inflammatory cytokines, which activates the patient's antigen-presenting cells. Those in turn activate the donor T cells, and at the same time, there is release of soluble inflammatory agents that further amplify that injury and inflammation. And all of that together culminates in what we know as graft-versus-host disease. But when the targets of that process happen to be cancer cells, we call that graft-versus-leukemia or graft-versus-tumor effect. So the holy grail of a bone marrow transplant is to have full engraftment, no graft-versus-host disease, and to maintain or even to enhance graft-versus-tumor activity. And since we said donor T cells are central to both of these processes, graft-versus-host disease and graft-versus-leukemia, how are their responses in recognizing the patient as foreign versus the tumor regulated? Well, that is where epigenetic modifications come into play. These are changes that sit on top of the DNA that can turn genes on or off without changing the structure of the DNA itself, the sequence of the DNA. And they broadly fall into three categories: DNA methylation, histone modifications, and chromatin remodeling. And they make good therapeutic targets because they determine the activity of genes. They are inherited as cells divide from cell to cell, but at the same time, they can be reversed. And our work here focuses on DNA methylation by DNMT3A. This is the main enzyme that catalyzes de novo DNA methylation, and we know by now regulates T cell function. It is an enzyme that is crucial in embryogenesis and hematopoiesis, and mutations in DNMT3A are actually very common in humans as we age in our stem cells. So several groups have actually gone back and looked to see what happens if your donor happens to be a little bit more on, the mature side and has a DNMT3A mutation in their stem cells. Well, those patients actually have an increased risk of GVHD, but a decreased risk of relapse, and actually end up having improved overall survival. So our hypothesis here is that donor T-cell DNMT3A regulates the donor immune response. And to test that hypothesis, we've used mice that lack DNMT3A in their T cells as donors in mouse models of bone marrow transplantation. The way that these models work is that we get the donor mice and we isolate bone marrow cells and splenic T cells, and then the recipient mice undergo radiation as conditioning. And the donor bone marrow cells will go on to provide immune reconstitution, and the splenic T cells will go on to cause graft-versus-host disease. And then we monitor these mice by survival, weight loss, a very well-established GVHD score. But at the same time for this project here, we have isolated the donor T cells after transplant and took a deeper dive into their methylation and gene expression status. So this is the phenotype that we observed. This is a survival curve. And in green are the syngeneic controls, meaning the donor mouse and the recipient mouse are identical. So they should do great. There should be no GVHD, so they do fantastic. Then we have an allogeneic control group, so wild type, aka normal donor mice into allogeneic F1 recipients. And there is some expected graft-versus-host disease, so the mice gradually die from graft-versus-host disease. But if the donor mice lack the ability to do DNA methylation in their T cells, the outcomes are much worse, associated with increased GVHD scores and increased weight loss. And there are many mechanisms behind that, so if you're more interested, come by poster 26 today, but there's not that much time today to go over all that, and I don't talk as fast as Dr. Kronk. We also then went on to look at graft-versus-leukemia activity. So here, again, the same, black 6 into F1, half-matched mouse. We cut down the T-cell dose because we don't want the mice to die from GVHD before they have a chance to develop graft-versus-leukemia effects. And at the same time, we add some tumor cells to the inoculum. And these tumor cells are transfected with a luciferase enzyme that lights up if the tumor is there. It's the same thing that makes fireflies light up. And then we monitor these animals. So you can see the syngeneic group, the donor immune system does not recognize the recipient immune system or the tumor as foreign. So these animals very quickly develop tumor and die. In the allogeneic group, the foreign donor immune system recognizes the tumor as foreign, so the development of leukemia is delayed. So there is some graft versus tumor effect there. But if your T cells of the donor lack DNMT3A, tumor control is much more efficient. And in turn, those mice go on to have improved tumor-free survival. So then to take a deeper dive here, we isolated the donor T cells after transplant, and we did whole genome bisulfite sequencing, which is considered the gold standard in methylation analysis. And we also did RNA sequencing to look at gene expression. And the aim of this was to intersect regions of differential methylation and gene expression between the different groups to identify signaling pathways that may be dysregulated in graft-versus-host disease that we can then go on to target. So you would expect if you knock out a major DNA methyltransferase, you might have just global DNA hypomethylation, but that is in fact not the case. Wild type and knockout T cells have very similar levels of mean methylation, but what we saw were very distinct peaks of localized hypomethylation, such as in the EGR3 gene here, which is a negative regulator of T cell activation. And all of those peaks actually made sense because they're involved in T cell differentiation and T cell signaling. And another example here of one of our top differentially methylated and expressed genes was CCR9, and we confirmed this in our RNA-seq data and flow cytometry data. And CCR9 is a chemokine receptor that drives T cells to the intestine, which is a major target organ of graft-versus-host disease. And in fact, single nucleotide variations in the CCR9 gene in the donors of transplant have been associated with increased graft-versus-host disease. So with this in mind, we reached out to a company and acquired a small molecule inhibitor of CCR9, and this inhibitor has already been tested in humans and has been proven to be safe, and we used it in our mice to see what would happen. And indeed, what we saw is that here in blue are the treated mice, and their white cells accumulate in the spleens and the lymph nodes. But in the intestine, graft-versus-host disease scores are improved, as is lymphocytic infiltration and apoptosis, a type of cell death. So then I always get the question, because DNMT3A mutations in humans originate in stem cells, so they don't only exist in T cells, what happens if the myeloid cells are mutated? And so we did those experiments as well, and there is a phenotype there. You can see again in the blue line that the mice do worse, but it's not as profound as in the T cell knockout model. And then another interesting thought here is, okay, we just showed if we take out the enzyme that does DNA methylation, the mice do worse. What if we take out the enzyme that does the opposite? Because TET2 is an enzyme that catalyzes DNA demethylation. It goes and removes the methylation marks. That should be the opposite phenotype, right? It should be better. Except it's not. It's very similar. And the explanation behind this is probably that even though these two enzymes have different functions, they probably have similar programmatic goals in the cells. And so to conclude, transplantation is a life-saving option for many children with cancer. Graft-versus-host disease remains a significant problem, and augmenting graft-versus-tumor activity to prevent relapse is also a critical need. DNMT3A regulates alloreactivity post-transplantation, as evidenced by human and animal data. And this approach here can uncover new potential targets for regulating post-transplant immunity to limit immune escape and improve outcomes. In future directions, we're all excited about using this approach as a discovery tool, not only for uncovering new targets for GVHD, but mainly for finding the pathways that differentiate deleterious graft-versus-host disease from beneficial graft-versus-tumor effects. We are looking more into the myeloid DNMT3A deletion phenotype, as well as the TET2 impact on graft-versus-host disease. Expanding into looking at the impact of recipient DNA methylation and transplantation outcomes, and then expanding into other epigenetic modulators, such as microRNA targeting, and Dr. Dionisio, who is in our lab, has a poster here today as well on that. So with that, I want to thank our mentors, collaborators, lab members, all the funding sources, including CCF, of course, and patients like Kate and Nicholas who are here today and inspire us to put in the hard work.
So I first want to say that when I proposed the idea of a symposia like this to CCF, one of the things that I said to them was that it's really important for the trainees and people who are moving through their careers to be able to explain their science to not only other scientists, but to people who don't have a scientific background. And in past years, I have to say that when we first started, I did a very active interaction with people who were going to be speaking at this to convince them and to help them find ways to speak in a way that others would understand. You have done an amazing job of that, and it's to be commended because I think your introduction brought everybody along with what it was you wanted to say. So for that, thank you, and it's really appreciated because I think the more people that understand what we're doing in medicine and science research, the more likely is people are going to be supportive. So that's wonderful. It also means that you'll get ideas from people that we wouldn't otherwise get because people understand. So I wanted to ask you, first question, so think about it. If you guys have questions, get ready to raise your hand, but I get to ask the first one. So you noticed that whether you methylated or stopped demethylated, you had similar phenotypes. So both of these enzymes are part of complexes of proteins where you have not just a single protein, but many, many proteins all working together to do these things. Is there any clues from the other proteins that are interacting in these systems that maybe the methylation itself may or may not be the right answer, but there may be other proteins involved in the other epidemiologic processes that might actually be the ones you need to go after?
That's such a great question, and it's honestly something I was thinking about last night as I was reading a paper on the interactions of these two enzymes, because they work on methylation or demethylation, but it's very possible that the real cause of the phenotype has nothing to do with methylation. So it's possible. The answer is I don't know yet, but we're excited to look into it, and very much a possibility. Yes.
All the way over in the corner here, and then anybody on this side?
Yeah, Yiouli, I thought this was an awesome talk. Thank you. I have a quick question for you. I'm sure you're aware of the data that came out, Steve Gottschalk's and Ben Youngblood's lab, where they showed that DNMT3 knockout enhances CAR T cell function. So I wanted to know if you've looked further into the T cell phenotype in
Yes.
your animals, if you have done any TCR sequencing, and if you noticed any differences in the stemness formation of these cells in your
Yes,
model.
absolutely. The Youngblood group has done a lot of this DNMT3A work in the chronic infection context and then in CAR T. And what they showed is that if one of the problems with CAR T cells is that they can work really well at first, but then they kind of like peter out, like they kind of get exhausted. So if you take out DNMT3A in them, because DNMT3A and DNA methylation is an enzyme that helps T cells naturally acquire these exhaustion programs in our body, if you take it out, these cells are much better at persisting in the patients, or the mice, I should say, and keep doing what they need to do. And so when we looked at our T cells, the phenotype, if you do gene ontology analyses, they're very similar to what the young blood group has noticed. Not exactly the same, because what methylation does changes from cell type to cell type and changes from context to context, but very similar in that the cells maintain a more effector-like phenotype and a more stem-like phenotype. So if you come by the poster, there's some of that information over there.
And again, your number is?
26.
Nothing wrong with a little shameless self-promotion, right?
Exactly right.
All right. Any other questions? Okay, well, with that, I'll thank you based on your slide. Ευχαριστώ.
Ευχαριστώ, bravo, thank you.
Yeah.